2 of chapter 12, we're going to start looking at the requirements of replication. So there's three major aspects to DNA replication that has to be very, very consistent for DNA replication to actually be successful. The first one is that you have to have a template of single-stranded DNA for DNA to actually be synthesized from. So you've got to have that template in place. The second requirement is there's lots of raw materials such as substrates and enzymes that are going to need to be present in the cell and going to have to come together and be assembled for this process to actually take place. And the third major requirement is that enzymes, as well as other proteins that their job is to read the template, actually has to be able to assemble in the DNA or assemble along the DNA for replication to take place. So three major requirements of replication: you've got to have your template, you've got to have your substrates in place, and you've got to have enzymes and other proteins in place for this to be successful. So in this figure, we can see that we've actually got a set of nucleotides that are connected to our sugar phosphate backbone. And again, this is really demonstrating the anti-parallelism of DNA. So we've got our 5' strand and our 3' strand here, and the red actually indicates the new strand of DNA that's being synthesized. So some things to remember: nucleotides during the replication process are always going to be added to the 3' OH end. So memorize that. Second of all, DNA is going to be made in a 5' to 3' direction. Two major pieces of information that you need to have a good handle on before you actually start to work through like how things are taking place. So with replication, like we see here, we've got our replication group and the 3' OH end, and this is where we're going to always be adding new nucleotides to. The two phosphates end up being cleaved off so that new nucleotide can be added on. And then in between the previous phosphate group and the new phosphate group is where our phosphodiester bond is going to form. So that's how we're actually bringing these units together. So again, nucleotides are always added to the 3' OH end of the new strand, and DNA is always synthesized in the 5' to 3' direction. Again, just taking a closer look at DNA synthesis, if you're going to start to see a lot of figures that look like this in your book. So let me orient you to this figure. The gray strands represent the template DNA, so the original DNA that for a certain chromosome in the nucleus. And go back. And this is going to be oriented. Our first strand up here at the top, our 5' end is over here at the top left-hand side, and the 3' end is over here at the right-hand side. So 5' to 3' direction. And then the second strand is oriented the opposite of that. So, and the opposite of the original template strand. So we've got a 3' end over here to the left side, and the 5' end is then facing the right side. So from these gray templates, we're actually synthesizing new DNA, and the new DNA is represented here by the red line, the thick red line. And as we can see, if we are synthesizing and using the top DNA gray template to synthesize the new strand, it's actually going to be done moving from right to left, because we are synthesizing in a 5' to 3' direction, and nucleotides can only be added to the 3' end. So this is what this process really starts to look like. If we've got either linear DNA or rolling sorry, not rolling circle, but theta DNA happening, then we've got two double-stranded pieces of DNA with bidirectional synthesis taking place. So that means that the new DNA is being synthesized on both of the template strands. So, sorry. So as the replication fork continues to get bigger, that's exposing more of the template for DNA polymerase to actually come in and start synthesizing new DNA strands. So those strands get longer and longer and longer. We can see down here at the bottom that we're starting to get chunks taking place. And this is because on the lagging strand of the DNA, you can only have the RNA polymerase will only come in and add a primer at certain areas. So we can't have continuous synthesis in this respect just because of the orientation of the template strands. And those lead to Ozaki fragments, and we're going to talk about those more in depth. So again, as our replication bubble gets bigger and bigger, we start getting these chunks of DNA that will eventually be glued together. And these chunks of DNA that we can see down here towards the bottom of the slide, these are going to be made up of the lagging strand. And so the lagging strand is discontinuous in nature. And so DNA synthesis is not continual like it is on the leading strand. Therefore, we get fragments of new DNA. And those fragments are going to have to be glued back together at the end of this replication process. So the leading strand is always considered as continuous, and the lagging strand is always considered discontinuous. And sorry, the actual definitions are not correct on this slide. So again, leading strand is continuous, lagging strand is discontinuous. Just flip-flop those. And the lagging strand is going to result in Ozaki fragments. And again, those Ozaki fragments on the lagging strand is where we have fragments of DNA being synthesized that aren't glued back together yet. That'll happen at the end of the replication process. So the theta model that we looked at is unique to bacterial systems. So this is what that looks like. If we've got one strand of chromosomal material and the original template was double-stranded, we can see as we start the replication process that it had come apart at the origin of replication, and then we started seeing synthesis of DNA material. So again, this is going to be continuous, so it's the leading strand, and then over here we've got discontinuous synthesis happening, therefore we're getting Ozaki fragments taking place, and that is the lagging strand. The rolling circle model looks like this, and rolling circle is really, really easy because you're only going to have the leading strand being synthesized, therefore there's no Ozaki fragments in the rolling circle model. And in the linear eukaryotic replication model, where we've got chromosomes like in humans or in livestock or in plants that are linear, and we have multiple copies of them because of the size of the genome, you're going to see one strand of DNA actually synthesize the leading strand, and one being synthesized as the lagging strand. So again, those lagging strands are going to have form Ozaki fragments that are going to be glued back together later. And as the replication continues to take place, the replication fork keeps moving forward thanks to DNA helicase, and the replication bubble keeps getting bigger until it meets another replication bubble.